I'm Connie Trimble. Thanks in advance for your attention. I'm a clinician. I take care of patients. So let me tell you some practical things about HPV. First, I have no conflicts of interest to declare. In fact, I'm just happy to still have a job. When I give a version of this talk to kids, sometimes I start this way. I ask them, how many of you have known someone who's had cancer? Raise your hand. Yeah. Okay. How many of you have known someone who had polio? This number of hands that go up is different than when I'm talking to sixth graders. But much of this is true because of vaccines. Cervical cancer is caused by a persistent infection with human papillomavirus. And although we've known how to screen for cervical cancer for over half a century, before we ever knew what the cause was, cervical cancer remains the second most common cause of cancer death in women worldwide. How can I, make those two sentences in the same breath is beyond me. We now have a preventive vaccine. We have many ways to screen for cervical cancer, but in my remarks, I'll focus on cervical disease, but I want you to keep in mind that HPV, we now know, causes cancers at many other anatomic sites in perfectly immune-competent people, and for disease at other anatomic sites, we have no validated screening algorithm. So it's actually a really big problem. I think it's one that we could get at in our lifetime, but here we go with biology. Okay, this is a slide I use for the medical students to drive home a point. Papillomaviruses are a family of viruses. They're double-stranded DNA viruses. They replicate, they have a really specific tissue tropism, so they replicate only in epithelium. And even different families have different tropisms for different anatomic sites. HPVs or papillomaviruses promote cell proliferation and survival rather than death. So they're really common. In fact, they're essentially endemic. In humans, there's four main groups. We think of one which causes genital warts, not so commonly cancer, another set which causes genital cancers as well as cancers of the head and neck, and then different sets completely which cause warts and skin cancers. The carcinogenic strains don't make warts that you can't even tell you have them. So here's an example of one mucosal lesions. Genital lesions, I think, is where most public awareness, and of course laryngeal lesions. On a bigger scale, from 40,000 feet, we know that 1 in 5 human cancers is caused in the context of a persistent infection. And this is a bar graph to show you the most common ones. And knowing the specific pathogens raises then the possibility of being able to manipulate a person's immune response to either prevent or treat disease. Most of the cancers associated with infectious pathogens occur in the developing world. We have viruses to prevent two of the most common ones, HPV and HBV. This is the HPV mantra from the point of view of a clinician. Normal cervix, we have several ways to screen. On the top panel, the pap smear, normal cells. The middle screen is a low-power photomicrograph of what the histology looks like. And you can also directly visualize cervixes. Now then, just about everyone gets infected or exposed pretty soon after they start having sex. Having said that, like other viruses that cause a lot of human disease, one of the reasons it's so common is because it doesn't do a darn thing to most hosts. And so you can't tell that you have it, you have no symptoms, and it takes quite a long time. It can take quite a long time for your immune system to clear it. So in the interim, you touch somebody, and so it's passed on. Having said that, in the percentage of people, maybe 10% we think, who can't clear or have a persistent HPV infection, that's the risk factor for the pre-invasive lesion, or CIN3. But we can screen for that also. And on the top panel is a pap smear. We can also do HPV testing. The middle panel is what the histology looks like. And again, we can directly visualize lesions. For those of you who have the biology gene, there's a very high nucleotide-plasmic ratio in the abnormal cells, and the screening direct visualization is not really high-tech. We put vinegar water on the cervix, and what that does is it dehydrates the surface cells, and abnormal cells have less glycogen, so they dehydrate faster. So then you look at it, and the abnormal cells turn white. We think CIN3 is the goal. Identifying CIN3 is the goal of most of our screening programs. We think that all invasive cancers arise from untreated CIN3s. Having said that, this continuum from normal to CIN3 to invasive cancer takes years and years and years, 10 years. So from a public health perspective, there are many, many options for preventing cancer. This is data on American adolescents. I made this slide before the last election. I have not been able to stop using it. HPV exposure does happen early and often. These are data on American adolescents self-reporting the onset of penetrative heterosexual vaginal sex. And I think there are two take-home messages. One is that boys and girls track pretty much together. And the other is that this surprises me, even though I've been doing this for a while, but by age 19, 3/4 of our young people report having had at least 4 partners. You betcha. So all of these factors add up to, we think that the lifetime risk of infection, at least once with a high-risk HPV type, is well over 80%. This is data that was published last summer on rates of uptake. You know, the vaccine, we think it would be a great way to prevent disease, but rates of uptake are disappointingly low. I have to say that, I'm sorry to say the disappointing part, but amongst eligible young people, the rates of uptake are less than 50%. And we did a study here in our system. You'd think that somebody who wanted the vaccine and got their first vaccine, well, okay, they actually really want the vaccine and they've chosen to get it. But the rates of completion in our system amongst vaccine initiators is 1 in 4. And I mean, those are people who got 1 out of the 3. So that raises a different kind of burden, I think, because it's our public penny that's paying for these really, really expensive vaccines. And if it's not going to be administered effectively, that adds more to the cost. Mind you, we need to maintain screening algorithms for a long time, too, because we don't know how long the benefit will last. The flip side, the cumulative risk of CIN3, pre-invasive disease we call these older women now, but that's over the age of 30. HPV screening is another way to potentially identify risk. With a one-time positive test for HPV 16 or 18, the cumulative risk for disease in women over the age of 30, you know, you don't want to be on one of those red or orange lines, basically. But having said that, okay, we have ways to screen for disease. How come people still fall through the cracks? We did a study here to try to figure that out. There are many steps a person has to go through in order to have disease identified. Okay, maybe they don't come in for screening. Maybe people don't come in for screening, and that's how come they get disease. Or maybe they come in and they have a healthcare visit and we don't screen them. Or they get screened and they have an abnormal test. Well, then they have to come see somebody like me. And they don't want to do that because I basically do everything that the dentist does, but down there. Okay, I can understand that. Or they come see me and they have an abnormal result and they run away again and they don't get appropriate therapy. So there are many ways you can see in real life that a person could be diagnosed with cervical cancer. Okay, if you had to, which way do you think is the most, is the reason that women in our system get cervical cancer? They don't get screened? The first one or the second one? You're right. We looked at all the new diagnoses of cervical cancer in our system. These were people who were, they had jobs, they had insurance, they had transportation, they had telephones, they had sort of low-grade medical issues for which they had seen a primary health care provider in the 3 years preceding their diagnosis. I mean, a little hay fever, a little sugar, a little depression, and we just didn't look. So this is a new soapbox that I have when I talk to the general internists. If you don't want to go down there, just send them to us. This graph is also relatively new. We have an increasing understanding that HPV disease, the incidence is increasing in other anatomic sites. I think it's safe to use the E word, epidemic, in terms of HPV disease in the base of the head and neck. HPV is causally associated with 25-30% of squamous cancers of the base of the head and neck. The incidence is increasing and will likely surpass the incidence of cervical disease in the developed world. As you can see from this graph, the incidence in males seems to be higher than in women, and I don't think the biology of that is very well understood yet. But the point I want to make is that screening is a luxury of a high-resource setting. We in the United States, we spend $6 billion a year chasing down abnormal pap smears. So in North America, where we have some money to spend on it, a diagnosis, two-thirds of women who are diagnosed with cervical cancer are diagnosed early enough so that we can cure them. But in a low-resource setting, a diagnosis of cervical cancer is essentially a death sentence. This is not an electoral map. This is a map of the incidence of mortality rates in the United States of cervical cancer. And you can see it's really geographically, the geographic distribution is really clear. It's up and down Appalachia and along the Mexican Tex-Mex border. Okay, I'm going to finish here with some slides about the differences between preventive vaccines and therapeutic vaccines, even though I'm preaching to the choir. So the idea behind a prophylactic vaccination is that you want to, infectious pathogens generally have specific receptors on their cellular targets, and the idea behind a prophylactic vaccine is to generate high enough levels of neutralizing antibody that basically recognize germ of choice, so that if your system is exposed to the germ, they glom onto it and basically just prevent the germ from entering the cellular target in the first place. Now then, for the rest of us who have all already been exposed to HPV, the prophylactic vaccines don't offer, we can't be protected from them. So what we work on across the street is immune therapies. In the case of HPV disease, the relevant biological event in high-grade lesions is that the HPV DNA has integrated into the host genome. Eventually, though, viral antigens will be exposed on the surface of infected cells. And so what we do is we try to devise strategies to make T cells that will recognize viral antigens and kill infected cells. The strategies for validating preventive as opposed to therapeutic vaccines are really, really different. For prophylactic vaccines, again, the idea is to inhibit infection in the first place. This requires huge test populations because you have to identify negative or naive populations first. Require many, many years for validation. What's your endpoint going to be? And the preventive vaccines have no therapeutic effect. Therapeutic vaccines, on the other hand, it's a different strategy. It's lesion elimination occurs via a T cell response. Antibodies are not germane. You can do this in relatively small test populations, i.e. somebody who's infected or somebody who's got a measurable disease, and viral clearance is a rapid, pretty quantitative endpoint. When I went to the FDA to get my first IND for immune therapies, they said, "Why do you want to like a therapeutic vaccine. We already have, you can cure disease. And I said, okay, well, if I told you that you had a really common infection in your parts, but don't worry, that a relatively trivial surgical procedure could cure 90% of lesions, and this is what your parts looked like after I got done, what would you think? And the body language was really interesting, because they all kind of went like this. I said, we think therapeutic vaccines are a great idea. This is my last slide. This is the incidence of polio in 1988, which I think is not very long ago. This is what the cervical cancer map looks like now. And with concerted efforts at vaccination, this is what it looks like in 2010. And I wonder if we can make HPV disease a history lesson for our kids.